there's a lot of controversy around genetic engineering. And so I want to talk a little bit about why it's important to have genetic engineering. With genetic engineering, we can produce proteins safer. We can produce more proteins. We're able to produce human insulin, which before this we used pig insulin, which wasn't as effective. We can make vaccines easier, and we can produce foods. We can produce healthier foods. Hold on one second. If I can get it to the next slide, you can see it. There we go. We can use eukaryotes or prokaryotes as the genetically engineered organism. So yeast have been able to be used in genetic engineering. We make genetically engineered plants and animals. These are termed transgenic. So we have plants that now can grow in environments where they couldn't grow before. We have plants that have toxins that are toxic to insects, so that they no longer, so farmers no longer have to worry about certain insects. We have plants that have certain vitamins added so that they places where these vitamins are scarce, people can get those vitamins. So example, beta carotene. We need beta carotene for eyesight. In certain countries where they don't have a lot of food items, they have a lot of rice, and they eat a lot of rice, which is good for carbohydrates, but they were missing beta carotene and people were going blind. Well, now we have this special golden rice that includes the beta carotene, and we have saved eyesight. So we're always making more, we're improving the world through genetic engineering. And so it's important to continue this. There are some drawbacks. I mean, there's always going to be some potential issues with genetically engineering anything, but I think the benefits outweigh the risks in general. And each individual has to be their own chooser on that. So you can choose to, well, you can't really choose to eat genetically engineered or not unless you're growing your own food. But even your own food may have already been genetically engineered when you bought the seeds. Who knows? So how do we genetically engineer things? So when we are looking for a vector, and so remember, in this case, our vector is the plasmid. When we are looking for a vector, we want one that has certain characteristics. So it has to have an origin of replication. It has to be able to carry cloned DNA. It has to have a restriction site, because otherwise we can't cut it. It has to have 2 selectable markers. So the first is some type of marker that selects for a resistance factor, and a common one is ampicillin resistance. The second selectable marker is to allow us to distinguish between the vectors that actually carry the gene and the vectors that don't carry the gene. And so an example would be a selectable marker that codes for LAC-Z. So the LAC-Z gene is a gene that helps in the production of enzymes to break down lactose. And so within this LAC-Z gene is our restriction site. And so basically, if we cut the LAC-Z gene and then we insert the enzyme, the gene that we care about, we're going to produce a broken LAC-Z gene, kind of like a transposon, except for we're inserting it ourselves, right? In this case, we're going to have non-functional proteins, and non-functional proteins on certain media will produce white colonies, while the functional protein, and I'll just erase this, which is not broken, is going to produce the gene that is supposed to be there, and those will metabolize lactose, producing blue colonies. So I made this to try and explain it a little bit more effective. So here we have a plasmid that has a restriction site and an antibiotic resistance site, okay? So within the restriction site is a gene that codes for beta-galactosidase, again, to break down lactose. And then here I just made a generic DNA sequence, and I'm saying it codes for insulin, okay? So we're going to cut both the plasmid and our DNA with the same restriction so that we have only the DNA sequence that we care about, and our plasmid are both cut with the same restriction enzyme. We're then going to allow them to anneal together. So we're going to put them together, and we do this by putting them into a solution and just letting them come together. So what happens is sometimes the plasmid is going to take up the DNA that we care about, but sometimes the plasmid is going to come back together without that DNA. Problem is, these are invisible. I mean, they're super small. Like bacteria, if you look under a microscope and you see them, they're super tiny, and they're 1,000 times bigger than what they really are. These plasmids are inside the bacteria, so they're even smaller. So we have to allow bacteria to take these in. And so what we typically use is E. coli. When we use E. coli, we have to make them competent. And we do this because E. coli are not naturally competent. We do this by shocking them in a process known as electroporation. A-T-I-O-N. Sorry if that looks bad. Okay. And so we add the plasmids and the bacteria together. What we end up getting is we're going to have some bacteria, once we've shocked them, that take in the plasmid with the gene of interest. We're going to have some bacteria that take in the plasmid, but there's no gene of interest. And then we have some E. coli that just didn't take in any plasmid. So these are three different types of E. coli now, right? We have E. coli 1 that has what we want, E. coli 2 that does not have what we want, but has the plasmid, and E. coli 3 that does not have what we want either. So the only way to separate them now is to grow them. So if we grow them on a basic nutrient agar, everything looks the same. E. coli look like E. coli, so they're all going to look identical. If we were to grow them on a media containing ampicillin, we're only going to get the bacteria that contain the plasmid. So this one obviously had no plasmid, right? And these two have the plasmid. What we tend to do is grow them on a media containing the ampicillin and lactose. And what that does, here again, no plasmid. And this one here contains no gene of interest. So that's just the plasmid without the gene. This is the one we want. And we know this because the gene was inserted into the restriction site that codes for beta-galactosidase. And because of that, we no longer can break down the lactose. All right, I'm going to stop this video. It's been 10 minutes, and we'll get into the next section in a few minutes. Bye.